Our first speaker today is Tina Annunziata. She did her medical degree and Ph.D. training at Georgetown University, where she also did her residency. Then she came to NCI and worked with Lou Stout in the metabolism branch on NF-kappa B signaling in multiple myeloma. And subsequently, she switched to ovarian cancer, and she now heads the clinical operations in the women malignancies branch. And she's a member of numerous societies, AACR, ASCO, and she's an associate editor of BMC Cancer. So she's going to lecture today on ovarian cancer in the genomics era. Tina.
All right, thank you very much. So thank you for the invitation to talk. I'm going to talk about the ovarian cancer and specifically in the genomics realm, as Terry mentioned. So what is cancer genomics? Genomics, as you know, is study of the genome, meaning chromosomes, gene expression, and a global analysis more so than individual entities. The genomics era, I would say, really started in 1959 when Newell and Hungerford studied the chromosomes to identify recurrent abnormalities, something called the Philadelphia chromosome in chronic myelogenous leukemia. So what is the Philadelphia chromosome? You can see here they looked sort of at a broad picture of what the chromosomes were, and they saw that patients with CMLs seem to all have, can you see my pointer here? Okay, seem to all have this little chromosome here that they call the Philadelphia chromosome because they were in Philadelphia at the time. And then it wasn't until 1973 that Janet Rowley discovered that this was actually a fusion between chromosome 9 and chromosome 22. And then in 1984, it was discovered that this chromosome fusion was actually at the breakpoint cluster region, which fused the ABL gene, which is a tyrosine kinase, to the breakpoint cluster region. And that caused constitutive activation of BCR of the ABL kinase to then drive the cancer cells. And then in 1996, so many years later, from 59, Brian Drucker discovered that you could block the ABL kinase using this drug, which eventually became approved for the treatment of CML. So that was revolutionary because in cancer treatment, they were actually targeting a specific mutation or fusion that was discovered by a genomics approach. So what is functional genomics? So those people didn't actually do a functional experiment until very late. In 1959, they just sort of did a observational experiment. Here, what is a functional genomics is to look for what part of the genome is actually functioning to cause an effect. What can actually transform cells into cancer? That would be a functional experiment. So here we're actually looking for driver alterations. You've probably heard that term before. What is driving the cancer? Is it a mutation? Is it a fusion event, et cetera? So in functional genomics, the first sort of major look into cancer was in 1981 with the discovery of the HER2 new oncogene. And the reason why it's called new, you may know, is because they used actually neuroblastoma that they put into mouse fibroblasts and saw what part of the genome would actually transform these fibroblasts into cancer. Then in 1984, Schechter et al. Discovered that new was very similar to EGF receptor. So that's when it came to be called HER2. And then in 1985, Cousins discovered that HER2 was actually present on chromosome 17. And here you can see the amplifications of this locus on these chromosome 17s. In 1987, Dr. Slayman et al. Discovered that HER2 was actually amplified most in some breast cancers. And here you could see the amplifications across some of the breast cancers. And then eventually you know that HER2 became targeted using Herceptin, which is a monoclonal antibody that can target the signaling of this protein. and is a pivotal treatment in the therapy of breast cancers that overexpress or have amplification of HER2. So how can we use genomics to study ovarian cancer to find out, do we have any drivers of ovarian cancer? So let's back up and talk about ovarian cancer itself. So ovarian cancer is the most lethal gynecologic malignancy in the US. It leads to 16,000 deaths per year. It's the 5th most common cause of cancer death in women. And that's because 70% of cases are diagnosed when the patient has at an advanced stage of disease, and less than 35% of these patients with advanced disease will be alive at 5 years. So what is advanced disease? We consider stage 3 and stage 4 to be advanced disease, and here you could see that most of the cases are diagnosed here in this advanced stage, and the survival, certainly of stage 4, is very poor, 5-year survival. Whereas if you could identify it very early, that's not frequently identified very early, but the survival would be very good. So what is the treatment for newly diagnosed ovarian cancer? First, it's complete surgical staging to make sure you're going for the correct stage. Optimal cytoreductive surgery, followed by or in conjunction with chemotherapy, and of course clinical trials at any stage of this. So the chemotherapy is very interesting. It's actually been a combination of platinum and taxane, so either cisplatin and carboplatin, or paclitaxel or docetaxel, and this has been the mainstay of therapy for decades. We have not really improved much upon that except for in recent years. Intraperitoneal, it's given intraperitoneal, which I'm going to talk about in a second, if it is stage 3 with an optimal reduction. So this is the brief history of where we've come with chemotherapy. In the 1960s, we only had alkylating chemotherapy agents, and no one survived beyond 5 years. In 1970s, it was the introduction of just the platinums, and we got about 5% of patients living beyond 5 years. In the 80s, we combined these with these, and we found, okay, maybe 15% of patients were now having long-term survival. In the 90s, we had the introduction of the taxanes, paclitaxel being the first one. And then we got survival up to 35%. So that was pretty big jump in 30 years to get 35% of women living long-term with this disease, with the combination chemotherapy. And then in the 2000s era, the advance has been to deliver that chemotherapy into the peritoneal cavity directly. And that's only applicable to a smaller subset of the women. So what is ovarian cancer from a biological standpoint? Most cancers are serous cancers, 80%, and that is looking like this top panel. But you can see that endometrioid, clear cell, and mucinous are also making up this type of this ovarian cancer that we call ovarian cancer, but they all look very different. So this is the clear cell, looks very different from the serous, which looks different from the endometrioid, etc. So, so why are we treating a disease that looks so different? Everybody's getting the same chemotherapy, even though they might be having a different disease. So we don't even actually know what the tissue of origin is. It's thought that maybe the serous cancers don't even come from the ovary. They actually start in the fallopian tube, especially in the patients with hereditary ovarian cancers. They might come in the fallopian tube and not actually from the ovary itself. Endometriosis can be potentially a source. So now we're talking even farther into the uterus. The endometrioid and the clear cell cancers might come from the endometrium that's spread retrograde back to the ovary and into the peritoneal cavity, which is also not directly from the ovary. And then those mucinous cancers, we don't know if they come from the glands or and from the cervix, going down past even further from the ovary, or if they come from an intestinal source. So there's still a lot up in the air that we need to learn about from a molecular standpoint. How are these cancers actually biologically different? And then we can hopefully get to better targeted cures. So of course, we hypothesize that increasing understanding about the biological and biochemical events underlying ovarian cancer might create new avenues for treatment. And the question is, can we use genomics to figure this out? So very early on, actually about a decade ago, in clear cell cancers, there was a very prominent study. To highlight here, we have clear cell cancers are a rarer subset of ovarian cancer, about 5 to 10% of cases, whereas the serous cancers, the high-grade serous cancers make up about 70%. Clear cell cancers do tend to have a worse response to the standard chemotherapy with cisplatin and paclitaxel. And as I mentioned, they can be associated with endometriosis in about 40% of cases. So back in 2010, so almost 10 years ago, this paper was published in the New England Journal looking at specifically genomic sequencing of clear cell ovarian cancers. So they started out by sequencing 18 clear cell cancers and one cell line in their discovery phase. And then they did targeted exome sequencing in 210 additional samples. And they threw in endometrioid and serous to compare there. And then when they found that discovery phase of validation, they looked at protein expression of their targets in another 455 samples. And what they found was that there was actually a recurring mutation or a bunch of mutations in the ARID1A gene. And so if you look at this, this is just showing you that these mutations occur throughout the locus of the ARID1A, and they have nonsense, they have insertions, they have missense mutations. So this kind of tells you that it's probably not an oncogene here, but maybe it's a loss of a tumor suppressor. Because if it was an oncogene, you would kind of have one activating mutation. But here we have mutations throughout the whole locus. So what is ARID1A? It's a member actually of the SWI/SNF chromatin remodeling complex. It has been found to be mutated in breast cancer and lung cancer. It's on chromosome 1p36, and it's actually deleted in 6% of cancers overall. So again, all of these things kind of suggest that it's a tumor suppressor gene where it has to be deleted or loss of function mutation. In this paper, they showed that there was actually loss of the protein. This is the protein of the ARID1A. It's called BAF250. So in 73% of the clear cell cases, there was loss of this ARID1A when it was mutated. There was loss of protein expression. So that, again, goes along with the tumor suppressor. There was also loss in some cases that did not have a mutation, so whether this is epigenetic silencing of the protein. Endometrioid, so this is clear cell endometrioid cancers, not as high, but still pretty important, but smaller numbers here as well. But virtually none of the high-grade serous cancers have loss of this protein. So this tells you now that we are finding a new biological difference between clear cell cancers and high-grade serous cancers. So to summarize, the ARID1A mutation was either lost, mutated or lost in over 40% of clear cell cancers, 30% of endometrioid, but less than 1% of serous. It's an unknown oncogenic mechanism, so you can't really target it like you would target an oncogene, but it has probably tumor suppressing properties, but it has unclear therapeutic utility. It might have a diagnostic purposes, but the reason why this was found was not a functional experiment. It was found by a sort of a screening experiment and not a functional experiment. Mucinous ovarian cancers is a less common subtype, even more so, even less common than the clear cells. Mucinous ovarian cancers, just so you see, with standard chemotherapy, have a worse overall survival than the overall population of ovarian cancers. So mucinous cancers also tend to respond poorly to standard chemotherapy. Gene expression analysis, again, this is an observational experiment and not a functional experiment. So they were looking at clear cell cancers, serous cancers compared to mucinous cancers. And actually in the gene expression profiling, mucinous cancers clustered together. So they have a different gene expression pattern than the serous cancers, either low malignant potential or high grade. Actually, based on this, which was published in 2006, in the cooperative group where we do clinical trials for ovarian cancer, the mucinous subgroup was taken out of those clinical trials, and we do separate clinical trials with targeted to mucinous patients compared to clinical trials that would include serous or other high-grade subtypes. So this was a classification that came out of a genomics approach. Oh, there you go. In mucinous cancers, what are the biological mechanisms? In a very small series, it was found that KRAS mutations were more common in mucinous cancers and also in low-grade serous cancers. So we're looking at serous cancers here, but we've now subsequently divided serous cancers into high-grade and low-grade because they do seem to have a different cell of origin, a different biological mechanism, and different mutation patterns. So low-grade serous are distinct from high-grade serous cancers in that they actually are more likely to have KRAS or BRAF mutations. It was found in 68% of low-grade serous cancers and borderline tumors, but none of the high-grade serous cancers. So now we're thinking these are actually coming from a different source, the low-grade versus the high-grade. And then this is just showing you graphically what I just said, which is that the serous borderline and the low-grade serous carcinomas actually have a higher BRAF or KRAS mutations, nothing in high-grade serous cancers, and very infrequently in clear cell or endometrioid. So what is the RAS pathway? You probably are pretty familiar with RAS pathway. It can be a driver mutation. If RAS is mutated, it can signal through RAF, which can also be mutated in separate cancers, signaling through MEK and ERK to stimulate gene expression, cell survival, and proliferation. So there was a clinical trial that was performed with a MEK inhibitor, so downstream of RAS. The thought was to block MEK with this drug called selumetinib. So they included 52 patients with low-grade serous ovarian cancer. They noted 8 responses, and 34 patients had stable disease greater than 4 months, which is considered somewhat of a benefit, not as good as a response. But the interesting thing that they found was that the KRAS or BRAF mutation was not really correlated with the response. So in patients, they had 5 patients who responded. Well, they had 7 patients who responded, and 5 of the 7 did not have a BRAF mutation. 2 of them had a BRAF or KRAS mutation. So the response did not correlate with the presence of the mutation, and the presence of the mutation in these 14 patients did not correlate with response. So what's going on here? We thought we had a mutational pathway that we identified. Well, we all know that RAS signals through the KRAS mutation can be signaling in a ton of different places, and we can't just block here to overcome that. So they are back to the drawing board with the RAS mutation, and we need better drugs to target more upstream, I think, is the answer. Okay, so high-grade serous cancer, as I mentioned, is the most common cancer, most common histology of ovarian cancer. So that was what was included initially in the Cancer Genome Atlas. The Cancer Genome Atlas looked for, obviously, molecular abnormalities that would influence pathophysiology, affect outcome, and constitute therapeutic targets. So in this initial study that was published in 2011, 489 high-grade serous cancers were analyzed for messenger RNA, microRNA, DNA copy number, and methylation. And then they did whole exome sequencing in a subset of those, 316. Patients had to be newly diagnosed, so no prior treatment. They had to have high-grade serous cancer. They had to have no prior treatment. And they had to have a companion normal tissue specimen. But this is actually pretty interesting. So they could have adjacent normal tissue, but they could also just have lymphocytes. So this will only really help in the DNA analyses and not the RNA analyses. So this was the initial finding. Interestingly, you could see here in glioblastoma, this is the copy number analysis, where red is shown in amplification and deletions are shown in blue. And you can see in glioblastoma, which was one of the first cancers that was included in the Cancer Genome Atlas, there's very distinct areas of amplification and deletion, and the rest of the genome is pretty quiet. In high-grade serous ovarian cancer, there's genomic instability, which is all throughout the whole genome. I mean, there's a lot of recurrent things, but it's not just one or two. It's pretty much every chromosome has amplifications and deletions. So then this became kind of a hallmark of ovarian cancer, of high-grade serous ovarian cancer, is this genomic instability, and how can we target that as a therapeutic mechanism. To look at the mutations, it was found that there was not a lot of recurring mutations. P53 was a hallmark, so P53 was found to be mutated in almost all of the cancers where it was sequenced. There were sequenced 316, and 302 of them had a P53 mutation. The next most common were the BRCA mutations, so BRCA1 and BRCA2. And you may be familiar with these as causes of hereditary breast cancer. It's also hereditary breast and ovarian cancer syndrome is what they call it now. So BRCA1 and 2. Then also the BRCA1 and 2 are involved in homologous recombination pathway. So they looked at other genes that might be affected in homologous recombination, which I'm going to talk about a little bit later. But you can see here that the BRCA genes were either methylated or mutated. The methylation is in green and the mutation, methylation is in purple and mutation is in green. So they could have, germline mutation or a somatic mutation, and then here is the methylation. So germline mutation is the purple, somatic mutation is the green, and then the methylation is in the blue-gray. So if you look at the BRCA, actually people with BRCA mutation tend to have a better response to standard chemotherapy than people without. So that was actually interesting prognostic finding. They also found other proteins or genes in the homologous recombination repair pathway that were similarly mutated. So they were thinking that this was a pathway that was affected in many ovarian cancers. And you could see that this was found in almost half of the ovarian cancers in this data set. These are some of the other major pathways that were altered, the cell cycle progression pathway, and this is the DNA repair that we already talked about. So what was the next steps after Cancer Genome Atlas? Were there new therapeutic approaches? Well, interestingly, there were 50% of cases that had the homologous recombination defect, and I'm going to talk a little bit more about PARP inhibitors that can be used to target homologous recombination deficient cancers. There were other commonly deregulated pathways like the RAS PI3 kinase pathway, the Notch pathway, and there were also a lot of aberrant genes or networks that were potentially targetable. So the idea here is that there's not one particular driver, such as we saw very early in the talk, where I talked about CML had that one driver through the ABL kinase. This does not have one driver. So we're thinking that we need to actually target network pathways in order to overcome chemotherapy resistance. Okay, so PARP inhibitors. This is definitely a hot topic in ovarian cancer. PARP inhibitors are most relevant in patients with BRCA mutations. So BRCA is actually not discovered by the Cancer Genome Atlas. The BRCA was actually discovered in 1990, when Mary Claire King and her group found that there was a recurring locus that was abnormal in families of hereditary breast cancer, breast or ovarian cancer. Then the cancer genome atlas actually showed us that beyond the BRCA mutations, there are other ways of altering the homologous recombination pathway. And that can be either through germline mutations, somatic mutations, methylation, or other mutations that affect this DNA repair pathway. Beyond this, people tried to come up with gene expression signatures, and actually RAD51 came up to be actually quite a strong predictor of homologous recombination deficiency. And you could see once patients with homologous recombination intact had a worse overall survival than patients with homologous recombination deficiency. This is actually a breast cancer, but the same holds true in ovarian cancer. So a lot of trials actually just presented this past week at the ESMO meeting in Europe. So we're trying to use homologous recombination tests to segregate patients to whether or not they should get PARP inhibitors. That's still experimental, but it's certainly up and coming. And this was just repeated in a cell line. So I've been talking about homologous recombination and PARP inhibition. So let me give you a little bit more background on the pathway that we're targeting here. So during normal cellular metabolism or environmental exposures, UV light, you can get single-strand DNA breaks. And so those single-strand DNA breaks are marked by the enzyme PARP, which stands for poly-ADP ribose polymerase. So that puts on a string of ADP riboses to mark that single-strand break. If you use a PARP inhibitor, that does not get identified. So you have your single-strand break that persists. In replicating cells, when you have the gyrase enzyme, you have to nick the DNA in order to untwist it, in order to get the replication going, the replication machinery in there. And that single-strand break gets converted to a double-strand break. So in a normal cell, the homologous recombination pathway is the best way of repairing a double-strand break. So in a normal cell, this would be fine. They would repair by homologous recombination, and they would be survive. In a cancer with a BRCA deficiency, this pathway is almost completely knocked out. So that cancer that has no BRCA cannot repair the double-strand break, and then it becomes overwhelmed with double-strand breaks and will undergo cell death. This was shown experimentally. Here's the PARP inhibitor Olaparib. It's an oral drug. And it has synthetic lethality in cell lines, at least with BRCA mutation. So here's the effectiveness. This is a dose escalation of the Olaparib, and this is the survival of the cell line in culture. Here's a cell line with wild type BRCA. Here's a BRCA2 mutation, and here's a BRCA1 mutation in the cell line. So you can see that BRCA1 and 2 mutations are dramatically more sensitive to PARP inhibition than cells with wild type BRCA. So we actually performed back in the early 2000s a phase 1 study combining Olaparib, which is the PARP inhibitor, with Carboplatin, which is a drug that can cause single-strand breaks. So we had one cohort where we had patients with BRCA mutation. We had another cohort with triple-negative breast cancer, which can have normal BRCA but might have a homologous recombination defect. And then we had another cohort with high-grade serous ovarian cancers, no BRCA mutation, but might have 50% of them might have the deficiency in homologous recombination. So we performed all of these, we had these 3 cohorts, and we did see interesting activity in the BRCA cohort. These are the dose escalation that we got to in the triple-negative breast cancer and in the Olaparib. And so just to show you the BRCA mutation cohort, we had enrolled 45 patients in this study. we did dose escalation. We had in the phase 1 was the 30 patients, and then the expansion was 15. So you'll see in the next slide, there's some patients who got lower than the maximum dose. The maximum dose in this expansion cohort was the carboplatin with an AUC of 5, which is the standard dose. Olaparib was given 400 milligrams twice daily. So these are the dose levels. So that's why I was explaining that we had patients on multiple dose levels, but even in the lower dose levels, we still had very good responses. So this graph is showing you the change in tumor size from baseline. And so 20%, actually 30% decrease is considered a partial response. And then obviously 100% is a complete response. Progression is more than 20% growth. So we had no one who progressed in this cohort, which is amazing. So these patients had previously received platinum alone, and they were resistant to platinum. We gave them platinum plus the PARP inhibitor, and nearly all of them had clinical benefit. So they all had either stable disease or a response. Of course, we had the most patients on dose level 6 because that was the expansion cohort, but even patients, like I mentioned, on dose level 1, 2, and 3 had responses. So this was actually a pretty amazing result, that we could have a therapy, a PARP inhibitor that was targeted towards BRCA synthetically lethal in cells with homologous recombination deficiency. We had no complete responses. That one that was really down far, it still had a little bit of disease left. We had 15 partial responses, which was 44% of the patients. We had 13 patients who had stable disease greater than 4 months. And then we had an overall clinical benefit rate of 82%. So that was very exciting. It was well tolerated in combination with the carboplatin, highly, highly active in BRCA deficient cancers. Of course, greater activity at the higher dose. But this is positive proof of concept, as I said, that the activity and tolerability of this combination regimen in genetically defined subgroup. Okay, so where can we go with new targets? We took a functional genomics approach. We were looking for actionable mutations. There's a lot of trials going on with actionable mutations. So how do we define that? People are using these commercially available tests, Caris or Foundation 1, to report these mutations that might be possible or unlikely to benefit. People are enrolling in basket clinical trials. For example, the NCI Impact will put patients in categories of therapies based on their mutational load or mutational profile. But typically in these settings, there's no functional link. So they observe the mutation, but they don't actually go back and do an experiment in the lab to show that that mutation is responsible for the sensitivity to a particular drug. So this was a review that was published in 2015. The word actionable depends in large part on the strength of the data linking the target to the targeted therapy. So for this basket trial designed to work, you have to have 2 key conditions. One is that the tumor must depend on the target pathway, and that the targeted therapy that you're using must reliably inhibit the target. And so these people correctly said that achieving both goals can be a matter of some complexity. So we thought that to define an actionable target, we actually needed a functional experiment. And so actually we went ahead and did functional genomics as one approach. The functional genomics approach that we took was based on a shRNA library that was published by Lou Stout in 2006 in Nature. He created an shRNA library that has a barcode. So these can all be put in a pool, and then you could pull out the individual clones or identify the loss of the clones using a barcode. In this particular setting, he actually targeted 2,500 human genes, and now it's up to I think 15,000. So we used this approach where you have the shRNA library, you infect the cancer cells, and then you either turn on the shRNA expression using an inducible construct, or you have the shRNA off. you look for the shRNA, you look for the dropout of the barcode in the shRNA on, and then that barcode will identify which genes were important. So we did this in 4 ovarian cancer cell lines. We used 2 serous ovarian cancer cell lines and 2 non-serous ovarian cancer cell lines. And we found that there were some genes that were common to serous and non-serous. And then we found some that were common to serous and some that were non-serous. So overall, in this sort of most common group, there were 63 shRNAs that represented 55 genes. And this is kind of busy, but these are the genes that were sort of most interesting. So we have followed these up in 6 additional cell lines using 2 different RNAi constructs in order to select the druggable targets. We're doing also functional focused screens in specific subgroups of ovarian cancer, and we have combination matrix studies that are ongoing. But one of the top targets was actually the gene for CHEK1. So this, we found that it was overexpressed in the Cancer Genome Atlas. It was overexpressed in nearly all of ovarian cancers, which supports the fact that we found it as one of our top gene in both serous and non-serous cancer. So it's generally an important gene in ovarian cancer based on its overexpression. And so we prioritized this as one of our top targets. What is CHEK? It is involved in the homologous recombination pathway. It's also involved in generic DNA cell cycle control. So what happens is when DNA is damaged, you have the DNA damage response. This signals through ATM to activate CHEK1 or CHEK2, which then goes in here and can block actually blocks the cell cycle at the G2M checkpoint. If this is amplified, you have more blocking, so you would have more time to repair genomically unstable genome that we already saw occurs in ovarian cancer. So we think that's why it's overexpressed, is because there's that genomic instability. The other thing is that cells with mutated P53 have lost this other checkpoint, which is the G1S checkpoint. So we think that in high-grade serous ovarian cancer, where P53 is knocked out for the most part, they really rely on this G2M checkpoint in order to repair any DNA damage that has gone on. Otherwise, they can't, and they would just go through the cell cycle with the mutated genome. And that could lead to those chromosomal abnormalities that we saw back in the Cancer Genome Atlas. So we found actually in our cell line screening that the high-grade serous cancers were actually more sensitive by several fold to the CHEK inhibitor than the non-serous cancers. So we do have a clinical trial that we're actually still enrolling to, and this is showing promising results specifically in the high-grade serous cancers that are the non-BRCA cohort. So this is very helpful because we now have the PARP inhibitors for the BRCA cohort, and we have potentially this CHEK inhibitor for the non-BRCA cohort. And I just wanted to highlight that this was found by a functional genomics approach and not just an observational approach. This is a study that is ongoing by my colleague, Dr. Jungmin Lee. She recently published this in the Lancet Oncology in February 2018. This is her clinical trial that found that the high-grade serous ovarian cancers without the BRCA mutation had actually a higher response rate than the ones with the BRCA mutation. So this is, again, very promising. Most of the patients are having some benefit, at least stable disease, and some even with response. These patients are still on study at the time of this publication. The gray bars mean platinum resistant. So these are patients who no longer respond to platinum therapy, no longer respond to chemotherapy, standard chemotherapy, but yet are still having pretty dramatic and long-lasting responses to this CHEK inhibitor. And again, she showed that patients, this is showing how long they've stayed on study. These are the ones with the documented response, but even those ones without response have still stayed on with stable disease for quite some time, and now we're out even longer than this because this was 2018. So to summarize, ovarian cancer is not just one disease. Ovarian cancer has the epithelial and a non-epithelial components. We did not talk about the non-epithelial today. In the epithelial, we have the high-grade serous that's marked by p53 mutations and defects in homologous recombination pathway, and these are potential targets for therapy. Low-grade serous cancers are a different biology. They rely on KRAS and BRAF mutations. Mucinous ovarian cancers, also different, also have KRAS, maybe have HER2 amplifications, and we actually have another clinical trial that I didn't talk about today that we're targeting a different antigen that's specifically expressed in mucinous cancers of the ovarian cancer, of mucinous ovarian and other GI mucinous cancers. So that's really exciting. We have clear cell cancers, which are represented by the ARID1A, endometrioid as well, share a biology potentially coming from endometriosis. And ARID1A is targeted by angiogenesis pathways, which I didn't talk about here. So just to summarize with the functional genomics, we started in a functional genomic screen where we just kind of took fragments of the genome and threw it in. And more recently, we actually do a functional genomic screen using shRNA knockout to see what is required and what will kill the cells if it's lost. And if we put that all together, if we can do a functional genomic screen, identify a driver pathway, do a clinical trial, we will have hopefully more success in treating individual cancers. So with that, I will take any questions. Just let me identify here. This is Jungmin Lee, who I mentioned ran the clinical trial with, or is still running the trial with the TRACO inhibitor, and she also was heavily involved in the PARP inhibitor studies that I mentioned as well. Stan Lipkowitz is our branch chief, and this is the rest of our team. Translational sciences for the functional genomic screens are listed here, and of course my collaborators in other branches. So thank you.
Any questions? Perhaps you could comment on the administration of the chemotherapy. Is IP still the best way to administer it?
So IP therapy is still considered the best way, specifically in women who have that upfront optimal debulking of stage 3. So that's really the only place that it's been proven to be beneficial. Other studies have looked in like earlier stage or later stage and have not been able to reproduce that benefit. The idea with the intraperitoneal chemotherapy is difficult to administer because it can lead to toxicity. So people are not, it's not widely adopted except in that one specific instance. We're actually doing an interesting clinical trial where we're doing IP cell therapy that I didn't talk about here, where we put in CAR T cells or activated monocytes. So that's another approach that we're testing out. Yes?
Is there a mechanistic explanation why the BBR cells respond to a checkpoint, while the BRCA cells were also kind of susceptible for genomic and do not respond to checkpoint inhibitor, and maybe consider to use to combine PARP plus checkpoint on the BRCA?
So that's an interesting question. So we're not sure why the BRCA ones did not respond. One of the thoughts, there's two kind of major areas. So one thought is that when BRCA mutated cancers become resistant to platinum, it means that they've remutated their BRCA gene to now have a functional homologous recombination pathway. So that's a possibility that their BRCA mutation has undergone a second mutation that restores the reading frame, and then they have now functional. So they're no longer having homologous recombination deficiency. That's one thought. The other thought is that there is there's other defects in the cell cycle pathway in the like cyclin E mutations in the non-BRCA mutation carriers, and that might be more gearing towards the check pathway, but that's not really proven at the moment. Those are thoughts. So what about combining the check inhibitor with the BRCA? I mean with the PARP inhibitor, I think that would be an interesting approach if we could do that in the future. Yeah.
Okay, that'll do it.
Okay.
And we'll take a little break here until our
Great.
speaker arrives.
Thank you.
Thank you, Tina.
You're welcome.
And our next lecturer is Stephanie Goff. She graduated from Georgia Tech, got her medical degree at the University of South Florida. She did a residency at Columbia University, and she did a surgical oncology fellowship combined with Dana-Farber Cancer Institute, Brigham and Women's Hospital, Massachusetts General Hospital, and then she came to NCI. She's going to lecture on immune checkpoint blockade. Stephanie.
Thank you. So this has been sort of a fun lecture for me to give over the last 4 years because as one of the slides that we'll get to at the very end shows, this has been a field that has sort of exploded. And in fact, although the Nobel Award winners for this year were just named the Nobel Award winners for last year, received their Nobels for this very topic. So let's just jump into a little bit about what I'm going to talk about. The first are the basics of immunotherapy. Then we'll get into a little bit about the mechanism of action of checkpoint blockade. We'll review a little bit of the early clinical experience and the discovery of immune-related adverse events because as a clinician scientist, these drugs actually really made us rethink about how we track drugs and how we dose escalate and how we create new drugs with the help of the FDA. I'll then go through some of the data that was the first clinical success of checkpoint blockade, which was in melanoma. And those are 3 of the drugs that we will talk about. They look like mouthfuls. They'll get a little bit easier as we go along. Ipilimumab, nivolumab, pembrolizumab. You may have seen these in television commercials. Those are otherwise known as Yervoy, Opdivo, and Keytruda. We'll talk about checkpoint blockade in cancers other than melanoma, and then just a quick diagram showing you where this might lead experimentally. So to start with, oncology, as you know, covers all of cancer. And traditionally, there have been 3 main ways to study cancer. Does anyone know what the very first way to treat cancer was? If you had a choice between immunotherapy, chemotherapy, radiation, or surgery, what was the very first one? What do you think we figured out first? How to cut things, how to burn things, or how to poison things? We learned how to cut them first. And actually, the very first mode of treating cancer was an operation, and it was actually back in 3000 BC. There's an Egyptian scroll that shows someone cutting a tumor off the chest. Probably not done up to the standards of today, and certainly without plastic surgery reconstruction. Next, do you think we figured out how to burn things or how to poison things? Burn. So Marie Curie, almost as soon as she figured out what radiation was, we tried to apply it to cancer. It's a double-edged sword. In fact, radiation, when it first came out, was used sort of as a toy. Shoemakers, shoe salesmen would use it to try to make sure people's shoes fit correctly. They would have these boxes, and the person would try on the shoes and put their foot in the box, and the shoe salesman would look down and see where their bones were in the shoes. And that was all fine and good until about 20 years later, all the shoe salesmen started getting thyroid cancer from the radiation exposure. So we no longer have that. But then because we figured out radiation could be so powerful, we started applying it to tumors. And then in the 1940s, there was work that happened first in Boston, and then it came here to the NCI to develop chemotherapy. And that started with young children with leukemia. And then the most recent pillar that we've added is immunotherapy. And the first immunotherapy got approved in 1992, and that was low-dose interleukin-2 for renal cell cancer. So it's only been about the last 3 decades that we've actually contemplated the thought that the immune system could play a part in cancer therapy. So we think there are 3 different ways to try to get the immune system to go after cancer. And the first, if you think about it, you've got a lot of T cells floating around in your body, a lot of immune cells, And the first way is to just to stimulate all the immune cells in your body, what we call a non-specific stimulation of immune reactions. And we can do this in two different ways. One is to give a stimulus that turns on all of the T cells in your body. And the other way is to turn off all the things that are holding back your immune system. And that is checkpoint blockade, which we're going to get into in a lot of depth in this lecture. But another way, if you think about those cells that live in your body as just sort of, elementary school cells, then another way is to teach those cells how to do it. And we do that with vaccines. So this could be like the high school version of T cells. They've been taught either with active immunization with either a peptide or RNA or a whole tumor. They've been taught how to see the cancer with the thought that then they can go out and attack it. The third would be the college type T cells, because we've actually taken them out of the body, done something to them in the lab, educated them as best we can, and then give them back. And that's adoptive immunotherapy. That's actually more my niche area, is what I do clinically. But what we're going to be talking about today is checkpoint blockade. So what are some of the cells of the immune system? This is kind of basic biology stuff. We've got two different immune systems. One is the innate immune system, which allows us to react very rapidly to anybody who's got a bad allergy, worried about anaphylaxis. That's part of your innate immunity. The second part of that, the anaphylaxis part, is actually the adaptive immunity. And what we think is the best way to attack cancer is actually part of the adaptive immunity, and specifically T cells. And that's what this checkpoint blockade primarily works on. So how is a T cell born? We've got lots of T cells, about 4 times 10 to the 11th. If you think about a thimbleful of blood, you've got about 1 million cells, million T cells in every thimbleful of blood. And we have this repertoire of T cells that circulate through our body. And when we're young, and even probably most of us in this room still have a working thymus, at least some remnant of it, and the cells go through there. If a protein is present in our body and creates a very strong reaction to this T cell, the thymus will actually destroy it, because you don't want your cells attacking your own body all the time. That is where autoimmune diseases come up. If a cell gets a very, not a signal at all, it will also get killed. It's those cells that are in the middle that make their way out into the world to sort of try to protect our body from things that we get from viruses specifically. So how does a T cell get activated? It's got a number of different signals. It's not just a one-step process. As you might imagine, anything that's in the body has multiple ways to get activated and multiple ways to slow it down. So we've got two different kinds of T cells. We've got a CD8 T cell, which is traditionally known as a cytotoxic T cell or killer T cell. We've got a CD4 T cell that's known as a helper cell. I'm sure you guys have known as you've gone through your career, everything's not as simplified as it is when it's first taught to you. So just know that CD8s and CD4s have far wider roles than just being helper and being killer. But for now, we're going to focus on that. What happens on the surface of a cell is that there is a histocompatibility complex, MHC, and that's sort of like your white blood cell type. And it determines what your body can see and can't see. Because as things are getting chewed up within your body by the proteasome, being placed on these MHC particles, coming through the Golgi, and then getting back out onto the surface, it's the specific shape of this MHC that will determine what piece of this peptide ends up on the surface. And then it's that combination of the shape of the MHC and the piece of peptide that's in the middle that the T cell can actually see. We'll get into this a little bit more later, but just to know that, for instance, if you're targeting things, say you want to target an antigen that's known to be on cancer, like KRAS or TP53, they may be present in 70% of all cancers, but because most patients don't share the same MHC, a T cell treatment that works for one may not work for the other, because it's the combination that's important. In another second type of T cell, the CD4, requires a different kind of MHC, or a class 2. And these are coming through, they're endocytosed, they're split into pieces, clip is required, and then they get into this groove of the MHC, and again, they're presented. What's important to know is that it requires the combination of the shape of this MHC and the peptide in between that can recognize a T cell. So it's that specificity that this one T cell, maybe out of 100,000 in your body, can see this particular combination. It's not going to be overwhelming, because if you had too many of them dedicated to one thing, you wouldn't be able to fight it. But everything that you got a vaccination for when you were young, or everything that you've been exposed to, all the viruses you've been exposed to as you've aged, you're going to have a population of T cells floating around, probably 1 in 100,000, 1 in 20 to 50,000, that can see each little piece of that virus. And what we're hoping is that we can exploit that for cancer. But the first part is that specificity. The T cell has to see its target in the context of MHC. If that doesn't happen, then all the other downstream stuff doesn't matter. The CD4 or the CD8 has to engage first. Once it engages, however, here's your T cell. CD3 is a marker for all T cells, whether they're CD4 or CD8. So once the signal 1 has been activated, all of these gray lines start to happen. The RAS pathway gets activated, the PI3 kinase pathway gets activated, PLC gamma pathway gets activated. But what has to happen is that there has to be a second signal. Now, if the second signal, you think about it, the T cell clicks together, then it's either got to click on or click off. If it clicks onto BTLA, PD1, or CTLA4, that T cell is going to get turned off. You see all these negative regulators of that interaction in the red. If it clicks on CD28 or ICOS, it's going to get activated. And that's how they activate all these pathways rather than blocking them. And then there are downstream pathways as well. Excuse me. So it's not only the specificity, that signal 1, it's the signal 2 that turns that cell either on or off. So it doesn't matter if it sees it, it's got to be clicked on or clicked off to make that cell work. And we call these the co-stimulatory molecules, which is a little bit of a misnomer because they don't all stimulate. Some of them regulate. But co-stimulatory molecules are the second signal on a T cell. And then the third signal, once you do get an activation of a specific T cell signal, then the presence of the cytokines that you're around will turn those into different kinds of T cells. And this is where I said it gets a little more complicated than just saying they're helper cells or cytotoxic cells. You can get different kinds of helper cells. You can get regulatory cells. And these all have sort of a different path depending on the cytokines that are present. So when that cell gets clicked on, depending on the milieu it's sitting in, will determine what sort of cell it becomes. So what these drugs do, they're actually signal 2 checkpoints. And the reason we have them is so that our bodies can promote self-tolerance. So the initial response to antigen will occur primarily in our secondary lymphoid organs. Those are the lymph nodes, the tonsils, the spleen. Peyer's patches are very specialized areas within the gut that have little patches of inflammatory T cells. And mucosa-associated lymphoid tissue, those are in the lungs. And what they do is they limit collateral damage. So when you get a splinter, the first thing that happens is that the immune cells rush to the side of that splinter. The splinter area gets red. If it gets infected, you'll start to get a little bit of pus. But then in order for that hole where the splinter went in, for that to start to heal, the splinter's got to come out. And then all of that inflammation that's there has to die down. So once it gets rid of whatever bacteria may have come in on that splinter, it's got to go away in order to let that wound heal. Any of you that have ever seen someone with sort of a chronic wound knows that it stays red, it stays inflamed, and it's because it can't limit that collateral damage. So for cancer immunotherapy to work, we have to create opportunities to break that tolerance. So a lot of the problem with cancer is that well, there are lots of problems with cancer, but one of them is that it looks like you up until a certain point. It's made out of your own DNA. It's the same cells that are in your body. It doesn't look foreign. It doesn't look like a virus. It doesn't look like a bacteria, so your body's not going to attack it. So we've got to figure out a way to break that tolerance. CTLA4 literally just stands for cytotoxic T lymphocyte antigen 4, is on the surface of T cells. So when this T cell that is naive or resting gets its first signal, so the TCR has seen its peptide in the context of MHC and gets activated because the co-stimulatory molecule gets clicked on, what happens to that T cell is that it starts to put CTLA4, excuse me, up on the surface of the cell. What that does, this oddly shaped piece over here, you can tell that it's sort of shaped like this one. It actually competes for that same ligand. So it will bind to the same ligand that has turned that on, and eventually will end up turning it off because it outcompetes. It actually binds more strongly here than it does here. So CTLA4 is transported to the surface in correlation to the strength of the CD28 stimulation, and it competes with a higher affinity for that CD80-86, which is the ligand for CD28. And it has a dampening effect, pardon me, on the downstream processing. CTLA4 is constitutively present on a special class of cells we call T regulatory cells. So T regulatory cells sort of exist to find T cells, other T cells, and turn them off. And this was actually first described by Jim Allison, who won the Nobel last year. His co-recipient, whose name I always mess up, is a Japanese scientist, pardon me. But he was discovering at the same time PD-1, programmed death receptor 1, which works in a similar fashion, in that when you've got a T cell here that's been turned on and it's hit its co-stimulatory receptor, it actually goes out to the tissue where the inflammation is happening. That inflammation on that tissue, where that splinter goes in, that tissue is going to start upregulating PD-L1 or PD-L2, the ligands for PD-1. And because that tissue starts to express it, it's putting out a, you've done your job, go away now, sort of signal to the T cell, and it starts to turn that T cell off. So a primed T cell is heading to the peripheral tissue to engage a target, and once it gets activated, it begins to express PD-1. The inflammation present in the tissue promotes upregulation of the ligands of PD-1, and this limits collateral damage during cell-mediated destruction. So it's why your hand doesn't start to destroy itself when it's trying to get rid of the splinter. Those cells get there, they do their job, and then they turn off again. So what's the role of PD-1, PD-L1 in cancer? Well, it works in two different ways. The first way is that there are some cancer cells that the way that they become cancer cells is they actually turn on the processing that upregulates PD-L1. So there's something about what makes them cancer that also makes them express PD-L1 on the surface. This is true of Hodgkin's lymphoma. The Reed-Sternberg cell of Hodgkin's lymphoma will overexpress PD-L1. And various cancers to a certain degree will express PD-L1 anywhere from 1 to 40% on their surface. So when that happens, going in, the T cell even if the T cell can see the cancer, it's going to get shut off right away. And then there's the adaptive immune resistance, where a tumor cell is seen by a T cell, and because that T cell starts to inflame the area around it, that interferon gamma that's released by that T cell to do that upregulates the STAT-JAX pathway, which upregulates PD-L1, which starts to turn it off. So our body, just like every other system where you're talking about sensing oxygen, where you're talking about sensing carbon dioxide, it's this regulatory feedback loop. And this is just one for the immune system. So cancer cells can increase the amount of PD-L1 on their surface, and successful T cell tumor destruction can increase PD-L1 through upregulation in response to interferon gamma. So when we started thinking about, well, can we block these and see if we can get a cancer tumor response, where would you start? So where we started, and I use the royal we because I was still in residency, were tumors known to respond to other immunotherapy. So melanoma, there's an estimated 9,000 deaths per year in the United States. When someone develops metastatic disease, that is when that melanoma spreads to other disease sites like the lung or the liver, patients only have about a 20% 5-year survival. However, interleukin-2, which is one of those nonspecific stimulators that turn on all the T cells in the body, could actually cure people. So these are people who got this drug with metastatic disease who were expected to die shortly. They're now alive 10, 15, 20 years later with no evidence of disease. Now that response rate is low, it's 4%, but it's a number, it's not 0. So that was one of the first places that we started to look. Then the other is kidney cancer or renal cell cancer, again, about 15,000 deaths per year. Also has a dismal survival rate for patients with metastatic disease. And again, interleukin had shown that it could cure some of these patients. Now, we don't use the word cure very often. We never use it with chemotherapy, and it's only because we've had the opportunity to watch these people for 15 to 20 years and their disease has not come back that we feel comfortable using that word here. So when we started with melanoma and kidney cancer, we actually started the first trial of anti-CTLA-4, which was ipilimumab, in conjunction with Dr. Allison, who had done all the work in mice. And so we started a phase 1 trial using a monoclonal antibody and enrolled 14 patients. So it was a very small trial. It was phase 1 just to determine the safety. But in that small trial, we had 2 complete responders and 1 partial response. But the way that drugs normally work when you're testing them in a phase 1 is you start at a very low dose, and if you don't get any side effects at that dose, you go to a higher dose. And if you don't get any at that, you go to a next higher dose. But what we found was that we started to get these toxicities, dermatitis and inflammation of the skin and inflammation of the colon and inflammation of the liver, and rarely this inflammation of the pituitary gland called hypophysitis. And what we were seeing, though, was that tumor could go away. This is a nodule that's in the lung of a patient. This was pre-treatment. You can see that solid gray mass, and you can see here that mass is gone. But at the same time, what you can see here, this is the skin, and you've got an influx of CD8. Well, unless you're a pathologist, you probably can't tell, but there's an influx of CD8 cells here that caused a rash of this patient, as well as here, these are colon, biopsies of the colon, an influx of CD8 cells that caused a colitis, which caused severe diarrhea. And then here in the liver, it's surrounding, oh wait, there's colon again. And then this is liver. So we're seeing really bad side effects at the same time we were seeing responses. And so the first thing we did, because we were seeing responses, but because we were seeing toxicities, we actually reduced the dose. So we said, well, 3 seemed to be a little bit too much. We were seeing too many side effects. Let's go down to 1. So we did that. But what we were finding was that people who were getting responses were the same ones that were getting the side effects. So that was completely different with chemotherapy. Chemotherapy, people would randomly get side effects, and it had absolutely no correlation to whether or not they were going to respond to the chemotherapy. So with that in mind, and it was pretty significant. If you looked at all people that had a grade, so our toxicities are graded from 1 to 5, 5 being that the side effect is so bad that the patient died, and 1 being sort of mild, so 3 sort of in the middle. So anybody that had a higher than grade 3 adverse event had a 36% chance of responding in melanoma. And in kidney cancer, it was even greater. So no patient that did not, I'm going to use a lot of double negatives here, if you did not have a severe side effect, you did not get a response. And so it was felt, instead of lowering our dose, we were going to push the envelope a little bit and actually increase the dose and see if we knew that we would be getting more toxicities, but we thought that we might be getting more responses. And so we did a formal phase 2 intra-patient. So that means that we would give the patient a dose of 3. If they tolerated that okay, we would give them a dose of 5 the next time around, and then we would give them a dose of 9 the next time around, all within the same patient. And what we found was that the correlation held true. If you had a grade 3/4 immune-related adverse event, 14 out of 50 patients responded for a response rate of 28%. Even if you had a low-grade immune-related adverse event, 8 out of 36 responded, which is still 22%. But if you had no immune-related adverse event at all, it was very unlikely that you were going to respond, 1 out of 53 patients. The most common was enterocolitis. Again, that's in the bowel. And what we did was we could give steroids, and the toxicities would go away, but the response would hold. So we figured out how to manage these immune-related adverse events. And you can see going out here close to 9 years, this is something we take for granted now in 2019. But prior to 2011, you never saw a drug that had this shape to it, what we call the tail of the curve. If you look at chemotherapy, even the best chemotherapies, if they prolong life a little bit, they all drop down to 0. Everyone dies because the chemotherapy just isn't capable of tackling metastatic disease. And so while we have gotten very comfortable with seeing graphs that linger out to the side here that have a tail on the curve, about 10%, 20%, these patients are alive 8, 9 years after treatment. And so this was sort of ushered in the new era of immune therapy. So in melanoma, they developed 2 means of blocking this. The first, remember we talked about CD28 as a positive stimulator, CTLA4 is one of the negative regulators, and PD-1 is one of the negative regulators. Monoclonal antibodies that block PD-1 and block anti-CTLA4. And those are the names that I discussed with you a little bit earlier. If in your reading of the medical literature you see things that have MAB at the end, that stands for monoclonal antibody. The MU actually means that it's murinized. The LIZ means that it's murinized, and then the companies get to pick what the first part is. Dr. Allison, who started his career at San Francisco, said that he really, really, really wanted to call this ipilimumab, but they wouldn't let him do it. So as all things go, when you start to get good responses, people want to make sure, well, you know, that's the NCI. They treat people that come, they pick them very carefully. We want to make sure this is applicable to a bigger group of people. And so they did a huge nationwide study of a phase 3. So they actually randomized patients to either get or not get ipilimumab. And what they found was that you get an objective response rate in 7%, including some complete responders. And this drug, ipilimumab, now known as Yervoy, was given FDA approval for patients with metastatic melanoma in March of 2011. That survival data was updated. 3-year overall survival of 20 to 26%, the tail of the curve, which we talked about before. So the other drug, nivolumab, blocks PD-1. And the kind of plot that we're looking here now is called a spider plot. It takes the sum of the measurements. You'll see these frequently if you look at clinical trials. The sum of all the measurable lesions that a patient has at the beginning, and then it tracks them over time. So starting point is 0. This patient here progressed at their first visit. Their tumors got bigger, which is why the slope is upwards. Patients where the slope goes downward, anything below 30%, which is this dashed line here, is considered a response. And so you could see they didn't know in what cancers this might work. And so they tested in a number: melanoma, lung cancer, kidney cancer, prostate cancer, and colorectal cancer. And you can see melanoma had a 28% response rate. Lung cancer had an 18% response rate. Renal cancer, 27%. But prostate and colorectal, no responses at all. And they only had grade 3 to 4 toxicities in about 6%. So the toxicity rate was better than ipilimumab, and the response rate was about the same. So they did a bigger trial. This time they took, because ipilimumab had already been approved, patients had to have already gotten ipilimumab to be on this trial. And so you're talking about patients not only with metastatic disease, but patients who have already tried ipilimumab, and it has failed to control their disease. So they're now probably, of those 12 months that we think a patient with metastatic disease has, they've already spent about 6 of them trying to get ipilimumab. This type of plot is called a waterfall plot. So instead of showing the way this one did, showing things over time, if you were to take the maximum deflection point at any one of these for each patient, you would get a waterfall plot. So for patients whose disease completely goes away, they all sit down here at -100. Again, this dashed line is showing you the 30% decrease, which is what we consider a response. And then anything that goes up means the patients have progressed. But in nivolumab versus chemotherapy of choice, they had a response rate of 32% with only 4 complete responses. And on the basis of this, it was approved for melanoma, refractory melanoma, in December of 2014. They then wanted to say, well, why do we need to make patients go through ipilimumab if this is better? Let's try it for patients who haven't already been through ipilimumab. What was happening in the background, though, was that people had uncovered BRAF mutations and a drug that could target BRAF mutations. So any patient that had a BRAF mutation needed to go get that other drug, and so they were not a part of this trial. So they tested nivolumab, which is the monoclonal antibody we're talking about. Dacarbazine is a chemotherapy that does very little for patients with melanoma. It will make things get smaller, but for a very short period of time, which you can see here in the survival curves. So nivolumab is the yellow line, and you can see that it starts to flatten out. And dacarbazine, like every other chemotherapy we've ever studied, is on its way down to zero. And again, this is only 15 or 18 months. If you were to extend this out to 36 months, this hits zero. This hits zero at about 16 months, and this is still going at 30%. So objective responses with some CRs. You can see this tail of the curve here. So 5-year overall survival of 34%. Pembrolizumab is just a similar form, also targeting PD-1, but made by a different company. So one is based on an IgG2, and one is based, I believe on an IgG1, and the other is based on an IgG4 backbone. But again, this was for patients who were refractory to ipilimumab, and these two companies were sort of in a battle. Who was going to get the FDA approval first for each different indication? Nivolumab one for one, pembrolizumab one for another, but they're both, I'm sure you see them both on TV, Opdivo and Keytruda. I think they both spend about the same amount on their ad buys. So they did a phase 1 dose comparison. You could see between 2 milligrams and 10 milligrams, there's actually not much difference in the two doses. But again, you see this flattening of the curve, this tail of the curve, which we all got excited about. And again, they got FDA approval in September of 2014. Again, this is now randomizing versus chemo. And you can see the tail of the curve coming out. Then we wanted to see for first-line therapy. So again, ipilimumab had been completely approved. Pembrolizumab had not been at this point. So they randomized. And what they found was that pembrolizumab had about a 30% response rate, ipilimumab only about 12%. All with impressive survival curves, but you can see ipilimumab lags below the others. Ipilimumab also had a higher incidence of side effects. So these are the updated survivals that were published in 2016. And you can see we're out 2 years, 60% survival. So again, just to remind you, median survival for patients with metastatic melanoma was about 12 months 10 years ago. So this is sort of a cartoon of the different checkpoints that can happen. And these are the two approved antibodies. And you can see they work in different spaces. They don't overlap at all. So the question came up, can we combine these two to get a better response? So these are patients that were previously untreated. It was a phase 3 randomized control, one versus the other versus the combination, 945 patients, which is a big trial when you consider that's probably capturing about 10% of all patients with melanoma in the country. Number of patients that got a response, 44% with nivolumab, 19% with ipilimumab, close to 60% with the combination. That sounded great, but then the grade 4 adverse events, 16% with nivolumab, 27% with ipilimumab, but the combination, over half the patients had a severe autoimmune side effect. And so these are updated results that were presented at ASCO a few years back, but have not yet been published to the best of my knowledge, but shows minimum follow-up of 18 months. The orange is the combination therapy, the blue is the single therapy with nivolumab, and the green is ipilimumab. And the FDA approved the combination for melanoma in January of 2016. So we got to thinking, why is this working in melanoma? This chart is one that if you get interested in the genetics of cancer at all, you'll probably see very frequently. It measures the number of mutations present in cancers based on a percentage of the total amount of gene of exome in that patient. And so you can see it has medians here at the gray bars, but then it shows you the wide range. Melanoma has the highest median tumor mutation burden of all the cancers. Lung cancer is next, bladder, and so forth. And the thought was, if your body only sees things that are different, then eventually this cancer has created enough differences that it looks different enough that the immune system's going after it. So highly mutated tumors. So what are the other highly mutated tumors? Well, non-small cell lung cancer is highly mutated, as is bladder cancer. As many of you may know, the kidney sort of filters everything that the liver doesn't. And so every toxin, every poison that we put in our body ends up sitting in our bladder for a little while. So it has the opportunity to mutate things. There's a specific kind of tumor called one with a mismatch repair deficiency. So over the course of becoming a tumor, it's lost the ability to fix its DNA every time that it replicates. So if a patient has this at baseline, a germline mutation, they will develop Lynch syndrome, which is a multi-cancer syndrome. Or the tumor itself can develop a somatic mutation in one of these 4 proteins. They're starting to identify a few more, but these are the 4 main ones. And so every time that tumor replicates, its DNA breaks apart and it's supposed to get zipped back together with a perfect copy. When those perfect copies don't happen, these proteins help put it back together. So if the tumor is missing these, then those places where it gets irregular never get fixed. And that's how you end up with highly mutated tumors. So nivolumab was tested in non-small cell lung cancer. It had a response rate of 14.5%. And it was approved for refractory squamous non-small cell lung cancer based on these data. Nivolumab as well. Again, these 2 companies going back and forth sort of for the same drugs. Pembrolizumab as well. So all of these data are in your handouts that you have. We don't need to go through it one by one. But the overall thought process is the same. You get a response rate of about 20%. It tends to be durable. There are some side effects that can be ameliorated by steroids. And if you can get the patient through them, then they have a good long-term outcome. Anti-PD-L1 is just blocking the other side of this pathway, and it was this drug called atezolizumab in bladder cancer, which is highly mutated. So in patients that had upregulation of PD-L1 on their tumors, they had a 43% response rate. And it was approved for urothelial cancer on the basis of that. And you can see, even though this is short, at 1 year, you've still got a 40% survival rate, which is sort of unheard of for patients with metastatic urothelial cancer. Again, just another example of how it's working. And another example, they all look the same. The tail of the curve started to show up everywhere. So now this got really interesting because every drug that you've ever heard of gets approved for one cancer or another. So pemetrexed for mesothelioma, or Folfox for colon cancer, or interleukin-2 for melanoma. It always is associated with a specific kind of cancer. But what we found was that in mismatch repair deficient cancers, it didn't matter what the cancer was, as long as it had a deficiency of these proteins. And they did 3 parallel cohorts. They looked at colon cancer that had the ability to fix all its mismatch repair. So these were proficient. And then it looked at colorectal cancer that had lost the ability to fix its mismatch repair. And then it looked at other tumors that had lost their ability to fix their mismatches. And it's very striking in that there's only really one outlier. Almost every patient that had a mismatch repair deficient cancer, whether it was colorectal or non-colorectal, these are the blue and the black. Almost all of those patients saw a decrease in their tumors. Whereas the converse was also true. If you had mismatch repair proficient, you progressed. The drug did very little for you. It's very rare to see something that clean. And when they looked at the pathology, here you're looking at a mix of tumor and normal, just adjacent to the tumor. you can see PD-L1 is high on all the tumor cells. There's a little bit in the normal tissue, but you can see where the CD8 T cells have started to come in to the tissue right at the interface. Whereas when you've got a cancer that has mismatch repair abilities, it doesn't have all those mutations. Very little PD-L1 is upregulated. Very few CD8 cells are recruited to the tumor. And you can see here, each brown dot is a CD8 T cell. There just aren't that many of them. So the immune system isn't seeing that cancer the same way. So for the first time ever in May of 2017, the FDA approved a drug not for a specific tumor type. You could have any kind of tumor that you can think of. If it's missing these proteins, it's approved to get this drug. And it's the first time that ever happened. Like I've talked about, going back and forth with these two drugs, nivolumab followed very shortly after. Macrosatellite instability is another marker of having a high tumor mutation burden. It is sometimes considered synonymous with mismatch repair. It is not, but they are very closely linked. And so FDA approval has been given for those as well. You can see this was July and this was May. So they've been neck and neck pretty much on every indication that they've been going back and forth for. So in highly mutated tumors, it works. Melanoma, non-small cell lung cancer, bladder cancer, tumors with mismatch repair deficiency. What about other tumors? So in kidney cancer, Hodgkin's lymphoma, head and neck squamous cell, and Merkel cell. So in kidney cancer, again, you're getting this tail of the curve. And because of this tail of the curve, it got approved in November of 2015. This was stunning. These are patients with Hodgkin's lymphoma. So classical Hodgkin's lymphoma, look how many of these lines drop below 0 and how many of them drop below 50%. So 7 went into complete remission of refractory Hodgkin's lymphoma. These are patients that have already had a stem cell transplant and had their lymphoma come back, and patients that have already been given brentuximab and had their disease come back. So these are patients that traditionally there would be no further therapy for, and 7 of them are in complete remission. Again, pembrolizumab followed shortly thereafter with waterfall plot that looks similar. In head and neck cancer, again, we're getting similar results, and it also received FDA approval. Now, Merkel cell carcinoma is an interesting one in that it is caused by a virus. So the virus goes into the Merkel cell and creates an aberration or a mutation. And because the virus is there, T cells come there. And so those T cells are there, they just don't have the ability to work because they get turned off by the checkpoint blockade. So by giving these drugs, you allow those T cells to get to work. And 32% of the patients in this trial responded, and 8 are in complete remission, and it also got approval. So when we block the PD-1, PD-L1 pathway, you can see that there is a number of different response rates. All of these have been approved by the FDA. So what about some other cancers? So in breast cancer, this was just a few of the trials that were presented over the course of the last few years. And you can see that while it's not as robust as some of the other studies, if you were to look at patients that had high levels of PD-L1 expression with metastatic triple-negative breast cancer, you get a 44% response rate. Now, granted, that's only 4 patients out of 9, so it's a very small number. But still, the fact that they're not 0 means that there's some role for the immune system in that trial. So in fact, the combination of a PD-L1 antibody and chemotherapy was recently approved in metastatic breast cancer on the basis of this paper, showing that when you looked at the 379 patients that had PD-L1 positive tumors, chemo plus this atezolizumab had a response rate of 60% versus chemo with placebo of only 43%. And the 2-year survival was 17% higher with the addition of the monoclonal antibody. And so that was approved just this year in March. It's the first approval of a checkpoint blockade for metastatic breast cancer. When I first started giving this lecture, there were only about 3 indicated approvals by the FDA. This was the first year I gave this lecture. And you can see now, as the years have gone on, the number of different cancers that have received approval for various immune checkpoint blockades have come up. And that includes pembrolizumab, which we've been talking about, nivolumab, atezolizumab. Durvalumab is another PD-L1, avelumab is PD-L1, and cemiplimab is another PD-L1. So you can see it's broadened: esophageal, endometrial, breast, cutaneous, small cell lung cancer, primary mediastinal B-cell lymphoma, cervical cancer, liver cancer, stomach cancer. So they're all getting approvals sort of across the board. What's interesting from a trialist standpoint is that if you were to go to clinicaltrials.gov right now, well, 2 weeks ago, there were 2,250 trials targeting PD-1 and PD-L1, about 1,700 of them in combination with something else. And this bubble plot here is just showing you what they're in combination with. So 339 of those 1,700 are anti-PD-1, PD-L1 in combination with CTLA-4. 283 of those trials are in combination with chemotherapy. 114 are in combination with radiotherapy. 58 are in combination with chemo and radiotherapy. And then some of these are other checkpoints. So there's a whole list of combinations that are coming along. So these are the checkpoints. So you've got a macrophage that shows things to a T cell. You've got dendritic cells that show things to a T cell. And you've got the tumor that shows things to a T cell. Anything that's got a red arrow is a checkpoint. And so we're learning how to block them sort of one by one. And I've tried to finish up early to leave some time for questions. But I know I just threw a bunch of data at you. So hit me.
So when a patient comes to you, how do you decide which one of the approved drugs to give them?
So a lot of it will depend on each histology. Sort of has its own that's been approved. So patients with melanoma, if you want to give them the combination, then that's only been approved for ipi and nivolumab together. If you have a patient who maybe, if you've got a young, healthy patient that can take anything you can throw at them, you want to give them the best chance for a response rate possible. So you'll give them the thing that has the highest response rate, even if it's got a high side effect. If you've got a patient who's a little bit weaker, who may already have a history of autoimmune problems like IBD or some other things, you don't want to give them a drug that is going to flare that up. And so you'll pick something that's a little bit weaker. So it all just sort of depends on where the patient is in their profile. And if someone's coming to us for a trial, a lot of these depend on certain markers in their tumor. So you'd have to get some molecular profiling first.
Deactivation of T cells, only their toxic effect or also T cell proliferation. It's not whether the durable immune effect of the checkpoint inhibitor affect to actually allow the T cells to proliferate or further activity of immunity.
So it's difficult to measure proliferation of T cells in the site where you want them. So it's very easy to measure T cells in the blood. And I can tell you that T cell levels in the blood don't change over the course of giving one of these drugs. But you can't always biopsy the tumor. And so even in the early stages when we were trying to learn about this as best we couldn't often biopsy the tumor. What others not here at the NCI have done is that you can actually look, the repertoire of the T cell will change. And so things that may have some bearing on the tumor will increase. And so your T cell population, which right now is very polyclonal, if you were to map out all your T cells, they would almost all be different. Once you've given one of these checkpoint drugs, you'll get these little bursts of certain TCR clonotypes that become a little bit more predominant. So it's almost like if I were to take you right now, it'd all be all flat. But if you were to get the flu, because you haven't gotten your flu shot yet, you'd get this little spike of flu virus. And then as your flu got better, it would go down. These patients get a little spike of what we think are the cancer-associated T cell clones, but they actually stay up over time while they're getting the drug. So it's difficult to quantitate, but qualitatively we know that you see little changes like that.
I was wondering what you think the field is for making therapy more specific. Is there any way to specifically target tumor pieces, T cells, or anything?
So I think that people are trying to do that with combinations of the big concept right now is that if you give a little bit of chemotherapy, it will kill the tumor. And we know that chemotherapy isn't effective at killing all the tumor, but if it kills it a little bit, it may release antigens, which can then prime T cells. So sort of giving chemotherapy or radiation to a tumor gives you this little burst, which is almost like a vaccine. And so if you do that at the same time that you're giving these checkpoint drugs, it should make it such that the T cells can go to the sites of the tumor much more easily. But again, that's all theory that's being tested right now. Drugs like this are never going to be completely 100% tumor specific because they're going after all the T cells in the body. The side effects that you see are evidence of that. So, it's very common for women to get a condition called Hashimoto's thyroiditis, which is where the immune system attacks the thyroid. You can see thyroiditis in this because our patients will have T cells already floating around that know how to see the thyroid, but they've been kept in check by these checkpoints. And so when we give the drug to help with the cancer, we inadvertently release the brakes on these ones that know how to see the thyroid. And so because people are not mice, we're never going to get a perfect tumor-specific model with these particular antibodies. Using sort of what I talked about in the very beginning with the cells, sort of what I called the college cells that have been taken out in the lab, you can get very specific with those, but that is a very highly personalized, very expensive, very complicated thing to do. These drugs, you just go in, you get an infusion every 2 or 3 weeks. It's a relatively simple thing to do. It's easier for the patients. Yeah, and there's a group here led by Dr. Gully over in the NCI that is looking at a PD-L1/TGF-beta trap molecule. So it goes to the site and supposedly will only go to a site where there's a high level of TGF-beta. And that might make it a little more site-specific. But those are still studies in trials right now. You got a question in the back? Mm-hmm. Mm-hmm. There's always a question of whether you're going to get fratricide, because part of what the T cell axis does is upregulate PD-1 and within the T cell itself. And so if you get to high enough concentrations of it, you can actually have T cells killing each other. We feel that the doses that are used in clinical trials now don't do that. You can often push the envelope in murine studies because you can treat them with much higher levels, and so you often can push it to where you see that phenomenon, but we haven't seen, not that I would say everybody's looked as carefully as they should, but we don't often see fratricide or killing of other T cells in the human studies. My boss, Dr. Rosenberg, is at a lot of cancer meetings. And so and we've been known as a place that can push the envelope. He had developed IL-2, so it was, he didn't discover it, but he helped develop it clinically. And so he feels very strongly that our role as the NCI is to take promising things. If there are investigators that don't necessarily have the resources to get a trial started quickly. And so Dr. Allison, I think, recognized that we had the opportunity to do something quickly. He was working on his own study. He was purely a lab guy. He didn't have any clinical experience at all and didn't have a great, he was working on basic T-cell immunology. So he was never really even part of a clinical cancer group. And once he decided that maybe this could start working in cancer models, he started working with some folks that knew Dr. Rosenberg, and I think that's how all that sort of worked itself out. Uh-huh. Uh-huh. A patient with metastatic breast cancer? Yep, that was my patient. I know Judy very well. She's appeared on newspapers and stuff, so I'm not divulging any HIPAA-related information. She is now 4 years out, still disease-free. So when I first met her, she had reached a point in her metastatic cancer where she was in severe pain, almost drowsy from the point of all the narcotics she was taking. I did an operation, took out a tumor that was on her breast, and then we studied that in the lab. It took us about 4 months to find the right cells that we wanted to give her. We gave those cells back to her in December of 2015, and I just saw her last month, and she's doing great. So she, part of not only the pain, but she had developed such a large tumor mass in her left axilla and her left brachial plexus that she couldn't move her left arm. And she was a very active hiker and kayaker, and so this was really affecting her quality of life. She just circumnavigated Florida in a kayak last year. So one of these days, cellular therapy will become more mainstream, and we'll get it into TRACO. It's not mainstream enough yet to be part of the curriculum. Mm-hmm. Yeah. She had hormone receptor positive when she initially got diagnosed in 2003. When we resected the tumor that I resected for her TIL therapy, it had actually lost its hormone receptor positivity. So that particular deposit was triple negative. But because we didn't biopsy everything in her body, we had to presume at least some of them still had hormone receptor positive tumors there. So she's lucky that she had the 4 months. Yeah, that's why cell therapy is still very much a thing of the future. We use it right now as a tool to learn as much as we can about the immune system in humans. But it's not for everybody for that reason. And it's what I've spent the last 5 years doing, and hopefully we'll spend the next 25 years doing. So. All right. Thank you.